Hi everyone, this is Jeff Hoffman. In cell biology, we'll focus on how the molecules and the biochemical pathways give rise to the behavior and interaction of cells. I'll try to keep it relevant to the boards and emphasize things you're most likely to see on test day. And because I want to provide the best lectures I can for you guys, it'd be helpful for me if you can use the thumbs up and thumbs down buttons at the bottom of the player here to give me feedback. Alright, let's talk about cells. To begin this section, we'll talk about the phases of the cell cycle. The most general phases of the cell cycle are interphase and mitosis, where interphase is basically growth of the cell, and mitosis is its division. Let's start with interphase, which itself has three parts, G1, S, and G2. So what does a cell need to do during interphase? One of the main limiting factors of cell division is how fast it can grow, since it can't divide until it gets big enough. And what limits how fast a cell can grow? Mostly the amount of ribosomes it has, since cell mass is mostly dependent on the abundance of proteins, and proteins are made by ribosomes. This growth happens during G1, which is the longest phase of the cell cycle. This is also the only time that cells can exit the cell cycle to enter G0 phase, in which they are not dividing. Some cells in G0 are still capable of dividing when stimulated to do so, and can re-enter the cell cycle in G1, such as fibroblasts, hepatocytes, and lymphocytes like this one here. Cells can also leave the cell cycle during G1 to become terminally differentiated, such as neurons like these, or muscle cells or erythrocytes, which will never divide again. Once cells transition into S phase, they're committed to complete the cycle, so S, G2, and mitosis progress much more rapidly. What's an example of a tissue that has cells that are constantly proliferating? There's a bunch of examples, but one is the skin. And by the way, these cells that are constantly dividing will be hardest hit by anything that interferes with cell division, like chemotherapy. Okay, what happens during S phase? S stands for synthesis, and this is when DNA duplication occurs. Therefore, cells in G2 have twice as much DNA as cells in G1. In both cases, they have 46 chromosomes, which is 23 pairs of homologous chromosomes, or 46 chromosomes total. But in G2 phase, each chromosome is made of two sister chromatids, whereas in G1, each chromosome only has one chromatid. Mitosis is the shortest phase of the cell cycle and includes prophase, metaphase, anaphase, and telophase. This is when the chromosomes condense and separate, and one cell becomes two. After mitosis and cytokinesis, the single parent cell is now two daughter cells. So let's talk about regulation of the cell cycle. What happens if a cell cycle is not regulated properly? Well, this can cause cancer. Cells can become neoplastic and form tumors. Therefore, it's really important that there are checkpoints that control transitions between different phases of the cell cycle, so if something is wrong and the cell should not be dividing, it can put on the brakes. One family of proteins that regulate the cell cycle are the cyclins, and the cyclin-dependent kinases. Cyclin-dependent kinases are constitutive, which means they're always around in all phases of the cell cycle, but sometimes they're active and sometimes they're not. So what activates them? This would be the cyclins, which is why they're called cyclin-dependent kinases. The concentration of cyclins fluctuates over time, depending which phase of the cell cycle the cell is in. There are several different cyclins and they're present at different phases of the cell cycle. The cyclins will activate specific CDKs, and the CDKs will then activate other proteins to do whatever needs to be done during that phase of the cell cycle. For example, in S phase, they'll activate proteins to initiate DNA replication. By the way, as a preview to a later part of biochem, what does the kinase and cyclin-dependent kinase mean? This means that they phosphorylate their targets, and phosphorylation is often used to activate or deactivate proteins. Since the G1 phase can last indefinitely, but once cells transition to S phase they're committed to divide, the most important transition to regulate is the G1 to S phase transition. This is why tumor suppressor genes, such as retinoblastoma and P53, are both able to prevent this transition. So what's a tumor suppressor gene? It's a gene whose protein product has a role in preventing cell division, and thus plays a major role in suppressing tumors. Okay, so what might happen to a cell that would make it want to halt the cell cycle? One of the most important things is DNA damage. P53 is one of the most notorious genes for detecting and responding to DNA damage, which has earned it the nickname the guardian of the genome. If P53 notices something wrong with the DNA, it'll prevent the cell from dividing, thus preventing tumors from occurring. When DNA damage is detected, P53 will stop the cell cycle and initiate DNA repair pathways to try to fix the damage. If the cell is able to repair the damage, the cell cycle can restart. However, if it can't, p53 will initiate apoptosis because it's better for a cell to die than to risk developing cancer. Retinoblastoma, or RB, also prevents the transition from G1 to S phase, but works a bit differently. Basically, RB has a few sites that can be phosphorylated, but after mitosis ends, RB loses most of its phosphate groups. In this hypophosphorylated state, it is active and it's suppressing the G1 to S phase transition. Only when it's inactivated by adding more phosphate groups will it allow the cell cycle to continue. And guess what type of protein adds phosphate groups to RB to allow the cell cycle to continue? That's our friend the cyclin-dependent kinase. Tumor suppressor genes like RB and p53 follow the two-hit rule, which means that both copies of the tumor suppressor gene have to be mutated to allow neoplastic transformation. This is because if only one copy is mutated, the other one is sufficient to regulate the cell cycle and prevent tumorigenesis. By the way, p53 is so powerful that more than half of all cancers have loss-of-function mutations in p53. Alright, time for our first flash quiz of the cellular section. The tumor suppressor genes p53 and RB prevent the transition from the blank phase of the cell cycle to the blank phase. So these guys block the G1 to S phase transition, because remember, once you get past that step, the cell is pretty much committed to go through the rest of the cell cycle.